Welcome back to Nucleus NEET Academy. I am Kiran Kumar, your biology NEET faculty. Students, now today let us discuss about another concept from chapter Biomolecules, that is enzymes. Have you heard about the term enzymes before? Have you studied about digestive enzymes? Yes. So today we are going to discuss about these enzymes in detail. So what are generally these enzymes? In your chemistry, you might have studied about catalysts. What does this catalyst do? They will simply increase the rate of chemical reaction. They will convert the reactants into products at much faster rate. They will speed up the rate of chemical reaction, isn't it? So just like those inorganic catalysts, inside our body also there are certain catalysts which are nothing but enzymes which act as biocatalysts. They will increase the rate of chemical reaction. So what is the definition of enzymes? Enzymes are biocatalysts that increase the rate of a chemical reaction, and essentially these enzymes are present inside the living cells and they will increase the rate of the biochemical reactions which occur inside our body. Okay. Next, almost all enzymes are proteins except ribozyme, which is a RNA enzyme. Generally, almost all the enzymes are protein in nature. They are made up of proteins. But there is exception. What is that exception? Ribozyme. Ribozyme, it is not a protein. It is a non-protein enzyme. It is made up of RNA. That's why we also call it as RNA enzyme. Even though it is made up of RNA, just like protein enzymes, this ribozyme can also catalyze the biochemical reactions. And the enzymes are produced by every living cell in our body. Every cell in our body, every cell in the plant or any living organism will produce the enzymes. Okay. In Greek, enzymes means in yeast. In Greek language, the term enzyme means in yeast. So why they gave the term enzyme? Because the scientists, they isolated the enzyme called zymase, which was found inside the yeast, and that is the reason the name enzyme was given. And who gave the term enzyme? That enzyme term was coined by a scientist called Kuhne. Okay, and enzymes can work inside cells and outside cells as well. Enzymes are biocatalysts. They can catalyze the chemical reaction inside the cells. But if you remove the enzymes outside the cells, then also they are able to catalyze the same biochemical reactions, provided that they have ideal temperature and pH. Understood the concept? Now look here. All the enzymes have tertiary structure. All the enzymes will show their catalytic activity in their tertiary structure, and generally the enzymes are named with suffix ase. So in name of any enzyme, most of the enzyme, there is a suffix called as ase at the end of the enzyme name. Example, amylase. Amylase is an enzyme which will break down starch into glucose. What is the suffix? ASE, ase. Protease, another example. Protease also ends with ASE. Cellulase is another enzyme, ends with ASE. Similarly, chitinase. But there are certain enzymes which does not follow this criteria of ending with ase suffix. So what is example for that? Papain. Papain is a enzyme. Actually, it is a protease enzyme, a kind of protease enzyme which is obtained from papaya plant. It can break down proteins into peptones and amino acids. This is also an enzyme but does not end with suffix ase. Okay, there are few such enzymes which does not end with suffix ase. Okay, next, enzymes are sensitive to temperature and pH. They work at an optimum temperature, a narrow range of temperature and a narrow range of pH. If the temperature is above optimum or below optimum level, then the enzymes simply stop working. Similarly, when the pH is above the optimum level or below the optimum level, then also the enzymes simply stop working as catalysts. Understood the concept? Then let us discuss about the structure of enzyme. You know that the enzymes, they have high molecular weight and they are globular proteins. If you observe the structure of enzyme, it will have a three-dimensional structure. It will have a three-dimensional structure. Okay, you can visualize the shape of enzyme like this. So it is having a globular shape with several irregularities on the surface. There are what you call bumps on the surface, and there are several grooves on the surface. So it is highly irregular. Okay, it doesn't have a proper geometrical shape, highly irregular surface. So these depressions which you can find on the surface of enzyme, these depressions, we call them as crevices or pockets of the enzyme. So the enzyme will have several of these crevices and pockets on the entire surface. But there is one pocket which is very special. So that pocket is called as active site, otherwise called as catalytic site. What is the speciality of this active site? So our substrate molecule, substrate means here reactant. The substrate molecule can bind to the enzyme in this particular place called as active site. Okay, so it cannot bind in any other place. It can bind only in this particular place called as active site. So this is the pocket which will be called as active site. So every enzyme will have a active site, otherwise called as catalytic site, which will allow the substrate molecule to bind to it. Understood the concept? When the enzyme binds with the substrate molecule at active site, now the enzyme will get chance to interact with substrate, and it can manipulate the bonds present in the substrate molecule. It can either break the bonds in the substrate, or it can make new bonds in the substrate, so that the enzyme can totally alter the structure of our substrate and convert the substrate into a new molecule called as product. Understood? Now let us discuss catalytic cycle. Imagine this is an enzyme, this is a substrate molecule. Our enzyme is having active site, a pocket called as active site. When enzyme interacts with the substrate, E plus S, it gives rise to enzyme substrate complex. Enzyme along with the substrate is called as enzyme substrate complex. Okay. Once enzyme substrate complex is formed, then the enzyme will interact with the substrate. It will create bonds, or it will break the bonds present in the substrate. So because of which the substrate, it will go through series of transitional states. After going through series of transitional states which are temporary, finally the substrate molecule gets converted into product. Same way, there is another possibility. The enzyme substrate complex again can revert back into enzyme and substrate. So that's why I have shown here double arrows. It is reversible process. Formation of ES complex is reversible process. If enzyme is not able to interact with the substrate properly, again enzyme and substrate may form. Okay, so that chance is also there. So after series of transitional states, the substrate molecule slowly it gets converted into product. Now enzyme product complex is formed. Look here, the product shape is completely different. Here I have shown substrate in a shape of a box. Here I have shown it circular. That means the enzyme structure has been changed and has been converted into product. Okay. Once EP complex is formed, then enzyme and product, they will separate from each other. Right. Enzyme and product both get separated from each other, and EP complex cannot again revert back into ES complex. It is irreversible process. ES to EP complex is irreversible. EP to enzyme plus product is also irreversible. So note down this. Now, is this enzyme function is over? No. The enzyme again it can interact with another new substrate and again catalyze a new catalytic cycle. So like this, a single enzyme can catalyze n number of chemical reactions. Are you able to understand? So this is how enzyme can function as a catalyst. So during the entire catalytic cycle, the structure of enzyme is not altered. The enzyme by itself, it will not change its structure. It will only change or alter the structure and shape of substrate, but not itself. Understood? So copy down this, students. Students, with the help of a simple example of a chemical reaction, now let us discuss how efficient the enzymes are in catalyzing a chemical reaction. Let us consider a simple chemical reaction, that is carbon dioxide reacting with water to form carbonic acid H2CO3. So this reaction occurs outside the cells as well as inside the living cells. Okay. Imagine the reaction is taking place outside the cells in a physical environment in a test tube. So when the reaction is taking place, without support of any catalyst, 200 molecules of carbonic acid are formed per every hour. So this is the rate of chemical reaction. 200 molecules of carbonic acid, which is a product, is formed per every hour without any catalyst or enzyme. The same reaction, when it occurs in the presence of enzyme called carbonic anhydrase inside the cells, then the enzyme simply it will increase the rate of reaction. So carbon dioxide combining with water to form carbonic acid. How many carbonic acid molecules are formed? 6 lakhs. Literally 6 lakhs molecules are formed per every second when the enzyme carbonic anhydrase takes part in the reaction. So from this we can say that the enzymes, they can increase the rate of reaction dramatically, and it was found that the enzyme had accelerated the reaction 10 million times. The rate of reaction was accelerated to 10 million times. So this is how the living cells, they will use the enzymes to increase the rate of chemical reaction in order to maintain the living state. Understood? Now, the amount of product formed in unit time is expressed as rate. That is, rate is equal to delta P by delta T, where P is called product and T is called as time. The rate of reaction can be expressed as delta P divided by delta T, where P is the product and T is the time. Okay. Next, enzymes show turnover number. What is turnover number? The number of substrate molecules which can be catalyzed by a single molecule of enzyme in unit time is called as turnover number. So this turnover number can change from enzyme to enzyme. Okay. Once again, turnover number means the number of substrate molecules which can be catalyzed by a single molecule of enzyme, only one enzyme, in unit time. That is called as turnover number. Students, now we will try to understand the concept of lock and key hypothesis proposed by scientist Emil Fischer. Emil Fischer, in order to explain the substrate and enzyme interaction, he proposed this hypothesis. According to this scientist, the substrate fits in active site like a key in a lock. So here you can see this is the substrate molecule and this is the active site on the enzyme. If you observe the shape of the substrate and the shape of the active site are complementary to each other. Because of this complementary shape, the substrate can exactly fit in the active site like this. So because of which a proper enzyme substrate complex is formed, and once the enzyme substrate complex is formed, the enzyme is able to convert the substrate into product. Okay. So here, in order to make you understand clearly, I have brought a lock and key. Our scientist Emil Fischer, he compared the substrate to a key and a lock to an enzyme. So just like a correct key which fits in a lock like this, a correct substrate molecule can fit in an enzyme to form a proper enzyme substrate complex. And only when a proper key fits in a lock, then only it is able to open the lock. So only when a proper substrate is able to bind with the enzyme in the active site, then only the catalyzed reaction will take place. Then only the enzyme can catalyze the reaction. Otherwise, there will be no reaction. Understood the concept? Later, another scientist called as D. Coastland, he proposed another theory called as induced fit theory in order to explain the substrate and enzyme interaction. According to Coastland, the binding of substrate on active site of enzyme changes the shape of active site to fit the substrate. He tells that the active site of enzyme is not rigid. It is not having a definite shape. Okay, it is a flexible. Whereas the substrate is having a definite shape and definite structure. So when a substrate molecule, when a correct substrate molecule approaches the enzyme and it fits into active site, then the shape of the active site automatically gets adjusted according to the shape of the substrate. So because of which a proper fit of substrate inside the enzyme takes place. So that is why it is called induced fit theory. The substrate is inducing the enzyme to make a proper fit. Okay, the binding of substrate on active site of enzyme changes the shapes. Look here, here the shape is different in active site. Here the shape of active site has changed. Binding of substrate on active site of enzyme changes the shape of active site to fit the substrate. So this is how the enzyme and substrate interaction takes place according to D. Coastland. It is called induced fit theory. Okay. So once enzyme substrate complex is formed, then the catalytic group inside the enzyme is able to convert the substrate into product. Okay. Understood the concept, students? Copy down. Now let us understand the enzymes, how they can lower the activation energy of a reaction. So any type of chemical reaction, if you observe, there will be two components. One is the reactants and another is products. During a chemical reaction, the reactants react and form a product. During this process, the reactants require some energy which is called as activation energy. By taking the energy called activation energy, the reactants will reach a transition state, and after then the reactants will react and give rise to product. So this is how a chemical reaction takes place, and the same thing I have represented in this graph. So on the x-axis I have represented the progress of reaction. So towards right side there are products, and towards the left side there are substrates, that is nothing but reactants. Okay, on y-axis I have represented potential energy. Okay, so during the chemical reaction, the potential energy barrier should be crossed by the substrate molecules or the reactants. So what is the potential energy barrier? That potential energy barrier is nothing but the transition state. So this line represents the transition state. So in order to cross this transition state potential energy barrier, the substrate molecules or the reactants require some input of energy, some input of potential energy. Okay, so when I am supplying some energy to the substrate molecules, then the substrate molecules, they will reach the transition state, and once they reach the transition state, they can break the barrier of transition state and they will react to produce the products. So this is how a chemical reaction takes place. Okay, so here what is activation energy? So the additional energy which should be supplied to the substrate in order to reach the transition state. So this much of potential energy, which is needed by substrate molecules is called as activation energy. Here you can see a considerable amount of activation energy is needed in order to convert the substrate into product without enzyme. Okay, so this energy we are calling it as activation energy without enzyme. without an enzyme. Okay. Suppose in the same chemical reaction where the substrates are getting converted into products, if you are using an enzyme, which acts as a catalyst, then what will happen to the activation energy? Suppose the enzyme participates in the chemical reaction, then the enzyme will reduce the activation energy considerably. The enzyme would reduce the activation energy considerably. So the substrate molecules will cross the transition state by using very less amount of activation energy and eventually results in formation of products. Okay, so here this much amount of potential energy which you are seeing here, this represents the activation energy with enzyme. Activation energy with enzyme. So now have you understood how the enzymes are able to reduce the activation energy and help the formation of substrates into products by making them easily crossing the transition state, which is the potential energy barrier? Understood? So here I will explain you with a simple concept. Here I have a plain white paper. So this plain white paper, it is having a micromolecule that is called cellulose. Okay, it is full of cellulose. The cellulose, you know, it is a wonderful fuel. It can burn. Okay, when we ignite the cellulose, then it will burn to produce carbon dioxide and water vapor. So in order to make this paper to burn, oxygen is also required. Oxygen molecules are present around us in the air which we breathe. So oxygen molecules are available. But why not oxygen is not reacting with paper to produce carbon dioxide and water vapor? Simply the reason here is activation energy is not provided to the substrate molecules. Who are the substrate molecules? The cellulose present in the paper and the oxygen which is present in the air. Suitable activation energy is not provided. That's why the paper is not burning. Okay, suppose if I provide some activation energy to these reactants simply by heating. So I am lighting a matchstick. Here there is a fire and now I am igniting the fuel. So the moment I ignite the fuel, the paper, so the substrate molecules get activation energy and immediately the reaction takes place. Here you can see the reaction is going on between the paper and oxygen molecules. The paper is burning, carbon dioxide gas and water vapor is being released along with formation of ash. So this is how the reaction takes place. Okay, so from this what you understood, activation energy should be provided to the substrate molecules or reactants in order to make the reaction to occur. Understood the concept? Now here some more important points you need to understand here. All the chemical reactions have potential energy barrier. They have to cross that potential energy barrier. Then only the reaction will take place and products will be formed. The reactants, otherwise called as substrates, need to cross the potential energy barrier to form the product. The energy required to break this energy barrier is called as activation energy. So the amount of energy which is required to break this energy barrier, what it is called? It is called as activation energy. And what is the role of enzymes? The enzymes mainly lower the activation energy. They decrease the activation energy while forming enzyme substrate complex, and they increase the rate of formation of products. In an exothermic reaction, that means the energy is released during the reaction is called exothermic reaction. In an exothermic reaction, the potential energy of the product is less than substrate. Here you can see, if you see the potential energy of substrate and product is having lower potential energy when compared to substrate. So what does that mean? That means it is an exothermic reaction. When the substrates are converted into products, the energy is being lost, the energy is being released. So because of the release of energy, the products have less potential energy. So when products are having low potential energy when compared to substrate, then we can understand that it is a exothermic reaction, energy releasing reaction. Suppose if the products have high potential energy, if products are formed here in this region when compared to the substrate, then what does that mean? It is a endothermic reaction. In endothermic reaction, the potential energy of product is greater than substrate. Okay, then the enzymes do not alter the potential energy of either substrate or product. The enzymes are not going to alter the potential energy of substrate or product. Simply what they are doing, they are just reducing the activation energy from this much to this much. Understood the concept? Students, now copy down this. Let us discuss about enzyme types. In living organisms, you can find basically two types of enzymes: simple enzymes and compound enzymes. Simple enzyme, you can see here, it is composed of only protein component. Example of simple enzymes are urease and amylase. Compound enzymes. Compound enzymes are complex in nature. They are made up of two components. They are composed of protein component and a non-protein component. The protein component or protein part is called as apoenzyme. You can see here, this is the compound enzyme. This part which you can see here, this is the apoenzyme which I am showing in shading. So this part of the enzyme is called as apoenzyme. It is made up of protein. Next, the non-protein part of compound enzyme is called as cofactor. You can see here, this part, dotted part, is the cofactor, which is a non-protein part. When non-protein part, that is cofactor, associates with the protein component, that is apoenzyme, then a holoenzyme is formed. The holoenzyme is able to carry out the catalytic function. Suppose if cofactor is not available, then apoenzyme alone cannot catalyze the chemical reaction. Okay, examples of compound enzymes are peroxidases, carboxypeptidases. These are some examples of compound enzymes, and obviously these enzymes require cofactors in order to show their catalytic activity. Whereas our simple enzymes doesn't need any cofactor, any non-protein part in order to show the catalytic activity. That is the difference between simple enzymes and compound enzymes. Okay, copy down, students. Now let us discuss about the factors that affect the enzyme activity. There are basically 5 factors which can affect the enzyme activity: pH, temperature, concentration of substrate, concentration of enzyme, and inhibitors. So now first let us see pH. Most of the enzymes have an optimal pH range in between 5.5 to 8.5. In this range of pH, the enzymes show their optimal catalytic activity. Suppose if pH decreases beyond the optimum pH, then the enzyme activity is going to decrease. If pH increases beyond the optimum pH level, then also the enzyme activity is going to decrease. You can see here, this is the graph which represents the enzyme activity based upon the change in pH. On x-axis I have represented the pH, on y-axis enzyme activity. So this particular zone is where you can see maximum enzyme activity. So this pH zone, you can call it as optimum pH. When pH increases beyond the optimum pH, then the enzyme activity is going to decrease. You can see here. When the pH is going to decrease beyond the optimum pH level, then also the enzyme activity is going to decrease. So from this, what we can understand that the enzymes can catalyze the chemical reaction at their optimal pH. And what is the shape of the curve? Bell-shaped curve is obtained when we plot pH against the enzyme activity. Let us see some optimal pH of different enzymes. Amylase has a 7 pH, optimum pH. Sucrase 4.5, pepsin 1.5. Now let us see another factor which can affect the enzyme activity, that is temperature. you know that temperature is one of the important factors that can affect the enzyme activity. Almost all the living organisms will maintain some sort of body temperature. So that temperature will make the enzymes to catalyze the biochemical reactions in a proper way. Understood? So most of the enzymes have optimum temperature in the range of 20 to 35 degrees centigrade. Okay. But what happens when temperature decreases below 20 degrees centigrade, such as the temperature goes to freezing point? At freezing temperature, what happens? The enzymes become inactive. That means they will stop their catalytic activity. They will just stop their catalytic activity at freezing temperature, 0 and below 0 degrees centigrade. Okay. Once again the temperature is brought to normal levels, room temperature, again the inactivated enzyme will regain its enzyme activity. Okay, there is no problem in that. But what happens if temperature is increased beyond the optimum temperature, above 40 degrees centigrade? Above 40 degrees centigrade, enzymes denature irreversibly. That means the enzyme structure will be affected permanently. Because of breaking of hydrogen bonds and disulfide bonds, the protein, the enzyme protein will lose its tertiary structure permanently, and thereby it will not be able to catalyze the chemical reaction. Once the enzyme has undergone denaturation process, again if you bring the temperature to optimum levels, then the enzyme cannot become catalytically active. Why? Because enzyme already it has become permanently denatured. Okay. So now let us understand temperature coefficient Q10. Scientists found that the enzyme activity doubles or reduced to half by every 10 degree centigrade temperature change in either direction. If temperature increases by 10 degree or decreases by 10 degree centigrade, there will be enzyme activity which is going to double, or it may gets reduced to half. This is called as temperature coefficient Q10. Okay. So just now we have discussed about denaturation of enzymes, but there is one exceptional case. There is an enzyme called Taq DNA polymerase which is present in a bacteria called Thermus aquaticus. So this enzyme, it retains the catalytic power at up to 80 to 90 degrees centigrade. It is almost boiling temperature of water. So even at this high temperature also, the enzyme is able to retain its catalytic activity. So this is one of the exceptional enzymes. Understood? Now if you see the graph, when we plot the temperature on x-axis and enzyme activity on y-axis, here you will get a bell-shaped curve. So this range is going to be the optimum temperature range. When temperature increases beyond optimum temperature, enzyme activity is going to decrease. When temperature gets reduced below the optimal temperature, then also the enzyme activity is going to decrease. So here also we get a bell-shaped curve. Okay. So next we will discuss concentration of substrate. Now let us see the effect of concentration of substrate on enzyme activity. The two scientists, Michaelis, Menton, they did a wonderful experiment to find the effect of substrate concentration on enzyme activity. So let us suppose there is a beaker. We have got a beaker. The beaker is filled with few enzyme molecules. Okay. Right now there are no substrate molecules, so the enzymes cannot show any catalytic activity. The moment we add substrate molecule to the beaker, this substrate molecule is going to interact with any of these enzymes, with this or this or this. So with any one of the enzyme, the substrate molecule is going to interact and the enzyme is going to show the catalytic activity. So initially when we add some substrate molecule to this beaker, so here there will be a small increase in the velocity of reaction which can be detected. Then gradually when we increase the number of substrate molecules to 2, then 2 substrate molecules will bind with the 2 enzymes. then further there will be increase in velocity of reaction. Like this at a time 3. So the velocity of reaction will keep on increasing as we increase the substrate concentration, like this. Okay, but at one point when the number of enzymes equals to the number of substrate molecules, at that time we can reach the maximum velocity of reaction. A maximum velocity of reaction will be achieved at that particular substrate concentration. Further, if you increase the substrate concentration more than 3 here, okay, if more than 3 substrate molecules are available like this, the excess surplus substrate molecules cannot engage with the enzymes because there are no free enzymes in order to engage with this extra substrate molecules. So there will be obviously no increase in the velocity of reaction further. So this is the point where a maximum velocity will be achieved, and after that further if you increase the substrate concentration, there will be no increase, but the curve becomes flat. The curve becomes flat like this. So this is called hyperbolic curve. A hyperbolic curve is obtained when we plot substrate concentration against velocity of reaction. By increasing the substrate concentration, we will get this shape of curve. Okay, so what is this point? This is the point where we have achieved the maximum velocity of reaction. The maximum velocity of reaction Vmax is achieved, and this is the corresponding substrate concentration. At this particular substrate concentration, we have achieved a maximum velocity of reaction. Further increasing the substrate concentration, we are not able to push the velocity of reaction any further in upward direction. Understood? So that particular maximum point we are calling it as Vmax, maximum velocity. Okay. Now Michaelis-Menten, they found that half of the maximum velocity, this point, half of the maximum velocity Vmax by 2. From half of the maximum velocity, if you intercept this hyperbolic curve and again we intercept the x-axis, we will get a substrate concentration value. This particular substrate concentration is called Km. Km refers to a constant called as Michaelis-Menten constant. Okay, so Michaelis-Menten, they have first discovered this constant Km, that's why it is called Michaelis-Menten constant Km. Okay, what is Km? Km is the substrate concentration at which an enzyme catalyzed reaction attains half its maximum velocity. Okay, Km, it is the substrate concentration at which the enzyme catalyzed reaction will attain its half maximum velocity. That substrate concentration is called as Km. What is the significance of Km? Look here, by using the Km value we can predict the affinity of substrate towards the enzyme. If higher is the Km value, lower will be the substrate affinity to enzyme. If lower is the Km value, then higher is the substrate affinity to enzyme. Affinity means attraction. If lower will be the Km value, higher will be the substrate affinity or attraction towards the enzyme. Michaelis-Menten, two scientists also gave an equation here. The velocity of any enzyme catalyzed reaction can be calculated by using this formula. Velocity of reaction V is equal to Vmax into substrate concentration divided by Michaelis-Menten constant Km plus substrate concentration. Okay, so this is about the concentration of substrate and its effect on enzyme activity. Now let us see how the concentration of enzyme will affect the enzyme activity. Again, let us consider there is a beaker and we have filled this beaker with substrate molecules, and slowly we started adding enzyme molecules to this beaker. As and when we increase the concentration of enzyme molecules, more and more enzymes are going to interact with the substrate molecules. Automatically the velocity of reaction is going to increase, but up to a saturation point. Once the number of enzymes becomes equal to number of substrates, highest rate of reaction, highest velocity of reaction will be achieved. Afterwards, it is going to level out. It will not increase. Okay. Rate of reaction increases with increase in enzyme concentration up to a saturation point only. After saturation point is reached, there is no increase in the rate of reaction. Okay. Next, what are inhibitors? Inhibitors are also factors which can affect the enzyme activity. Inhibitors are a kind of molecules. They are a type of molecules. These molecules, when they bind with the enzymes, then they are going to affect the enzyme activity. The enzyme inhibitors bind to enzyme and reduce their activity by either slowing down or completely stopping the chemical reaction. So, the inhibitors, when they are present along with the enzymes, they are going to affect the enzyme activity adversely. They may either slow down the rate of reaction, velocity of reaction, or they may completely stop the enzyme catalyzed reaction. Okay. How to overcome the effect of inhibitors? We can overcome the effect of inhibitors by increasing the substrate concentration up to certain level. Okay. So, the next topic we will see what are the different types of inhibitors. Okay, copy down this, students. Now let us see the different types of enzyme inhibitions. First is competitive inhibition, non-competitive inhibition, and allosteric inhibition, also called as feedback inhibition. First, let us see competitive inhibition. So look here, in competitive inhibition, this is an enzyme. The enzyme has an active site. This is the active site. The active site allows the binding of substrate molecule. Okay, but look here, there is another molecule called as inhibitor I. The inhibitor molecule, it resembles the substrate in its structure. So because of resemblance, the inhibitor will compete with the substrate molecule for active site. Okay, so now the inhibitor, it will compete with the substrate and it will occupy the active site. Once the inhibitor molecule occupy the active site, then the enzyme is no more will be in a position to catalyze the chemical reaction. Okay, so the catalytic activity completely will stop in the presence of inhibitor at active site. This is called competitive inhibition. Now look here, when the inhibitor, it leaves the active site and it moves away from the active site, then automatically again our active site is vacant. Then our substrate molecule once again can attach to the enzyme and normal catalytic reaction will take place. So that's why competitive inhibition is always reversible. Okay, why it is reversible? The moment inhibitor leaves the active site, our substrate can attach to the active site normally and normal enzyme reaction will take place. Okay, so this type of inhibition is called as competitive inhibition. Example is malonate, which is the inhibitor of enzyme called succinate dehydrogenase. Here malonate, it acts as an inhibitor. It will compete with the actual substrate called as succinate. Succinate is actual substrate. The malonate is going to compete with the succinate substrate for the active site present on the enzyme called as succinate dehydrogenase. Okay, so what is succinate here? Succinate is the substrate. What is the inhibitor? Inhibitor is malonate. Okay, now let us see non-competitive inhibition. The name itself, it is there is no competition. Non-competitive inhibition. Look here, this is the enzyme. As usual, there is an active site. This is the active site on the enzyme for the substrate molecule. But there is another site called as inhibitor site present on the surface of enzyme. This site is dedicated for our inhibitor molecule. Okay, when the inhibitor molecule binds to the inhibitor site at this particular site, then the shape of the active site is going to change. As a result, the substrate will not be able to bind to the enzyme. As a result, the enzyme activity will stop. Look here, the inhibitor binds to enzyme and inhibitor site other than active site. Okay, so because of which the shape of the active site will change. As a result, the substrate is not able to interact with the enzyme, hence no enzyme reaction, enzyme catalyzed reaction. So this is an irreversible type of inhibition. So once our inhibitor binds to enzyme, afterwards there is no going back again. The inhibitor never will leave the enzyme. So that's why it is an irreversible inhibition. Example is cyanide. Cyanide on the enzyme called cytochrome C oxidase. So during cyanide poisoning, what will happen? When people they swallow cyanide, a poisonous substance, it will go and block the cytochrome C oxidase inhibitor site. As a result, this is the enzyme, crucial enzyme which is involved in ATP formation in mitochondria. Hence, the cells will be deprived of ATP and the person will be going to die in the case of cyanide poisoning. Okay. So why it is so dangerous? Because basically it is irreversible. Once cyanide binds to the inhibitor site, it is not going to take back. Okay, that's why it is a serious condition, right? Now, another inhibition is allosteric inhibition, also called as feedback inhibition. Look here. This is a kind of mechanism which normally occurs in all the cells to regulate the enzyme activity. Sometimes the enzymes will catalyze more substrate into product, excess product, which is actually not required for the cell. In that case, this allosteric inhibition is going to help the cell, okay, to reduce the enzyme activity. Look here, this is the enzyme molecule. The enzyme molecule is having the active site. The substrate can bind to the active site, but there is another dedicated site for product. Okay. Look here how this mechanism works. It is very beautiful. Look here. When substrate binds to the enzyme, the substrate will be converted into product. The substrate will be converted into product. Once a suitable amount of product is formed, the product molecule can go and attach to the enzyme at allosteric site. So the moment when product binds to the enzyme at allosteric site, the enzyme activity is going to stop temporarily. Are you able to understand? Once the product concentration decreases, then the product is going to leave the allosteric site and once again the enzyme will restore its catalytic activity. Once again it will start the catalytic activity. So in this way, this allosteric inhibition mechanism will act as a regulatory mechanism to control the enzyme activity. Look here, the end product of the enzyme catalyzed reaction binds to the enzyme at site other than the active site called allosteric site and inhibits its action. Example, glucose 6-phosphate. It is a product which inhibits the enzyme hexokinase. You remember that hexokinase enzyme is involved in glycolysis. Okay, glucose is converted into glucose 6-phosphate. Okay, here glucose is the actual substrate, glucose 6-phosphate is the product. When more glucose 6-phosphate molecules are available, then they will act as allosteric inhibitors of the enzyme. Okay, so these are the three types of inhibitors. Copy down the students. Now let's see cofactors and different types of cofactors. Already we discussed about the concept of compound enzymes. What are compound enzymes? When a cofactor, a non-protein part, will bind with apoenzyme, which is a protein part, it results in a holoenzyme, which is a compound enzyme. Look here, this is a protein component of enzyme called as apoenzyme. When a non-protein component called as cofactor, it binds with the apoenzyme, it results in formation of a holoenzyme. Okay, so the holoenzyme is able to catalyze the chemical reaction. Suppose if cofactor will separate from the apoenzyme, then what will happen? Then apoenzyme alone cannot catalyze the chemical reaction. That means a cofactor is going to help the apoenzyme in catalyzing the chemical reaction. Look here. What are cofactors? Cofactors are basically non-protein molecules which help enzyme in catalytic activity by binding to the apoenzyme. Okay. So see here the formula. Cofactor plus apoenzyme gives rise to holoenzyme. Cofactor is a non-protein part, apoenzyme is a protein part, and holoenzyme is a compound enzyme. Already we have discussed some examples of compound enzymes, right? Now let's see the types of cofactors. Cofactors, basically there are three types. The first one is the prosthetic group, second is the coenzyme, third is the metal ions. What is a prosthetic group? Prosthetic group is an organic molecule. It is organic molecule. The prosthetic group, it binds tightly to the enzyme. Okay. Example is heme in the enzyme called peroxidase. Here heme acts as a prosthetic group in enzyme called peroxidase. Coenzymes are essentially, they are vitamins. Okay. B complex vitamins. They are organic molecules. They bind temporarily to enzyme. Temporarily or transiently to enzyme. Okay. Examples FAD, NAD, coenzyme A, TPP, all these are derived from vitamins. Okay. Flavin adenine dinucleotide, nicotinamide adenine dinucleotide, coenzyme A, thymine pyrophosphate. These are all the coenzymes. Okay. Which act as cofactors. Next is metal ions. Metal ions, from name itself we can understand that they are inorganic molecules. They form coordination bonds with active site of enzyme and also with the substrate. Okay. Examples of metal ions which act as cofactors are zinc. Zinc acts as a cofactor for carboxypeptidase enzyme. Potassium for pyruvate kinase. Copper for cytochrome C oxidase. So this is about cofactors. Copy down, students. Now let's see enzyme classification. Enzyme classification system was introduced by an organization called IUB. IUB stands for International Union of Biochemistry. Already you may be knowing different types of enzymes which you have studied in human digestion, and also the enzymes which are involved in respiration process like glycolysis, Krebs cycle, and you may be knowing the enzymes which are involved in photosynthesis in dark reaction like Rubisco enzyme, phosphoenol pyruvic acid carboxylase, etc., etc. Right? So if you study the reactions these enzymes catalyze, all these enzymes which are present in the living organisms, they fall under the 6 categories. So IUB, International Union of Biochemistry, has classified all the enzymes which are present in the living organisms into 6 types. The first type is oxidoreductases. Next is transferases. Next is hydrolases. Next is lyases. Next is isomerases. Next is ligases. So total 6 types of enzymes. Now let's see oxidoreductases. The name itself tells that they are involved in oxidation and reduction reactions. They catalyze oxidation and reduction reactions. If you observe the mechanism, let's say there are 2 substrates, substrate which is in reduced S dash which is in oxidized form. Then S reduced form substrate is converted into X oxidized, S oxidized, and S dash oxidized substrate is converted into S dash reduced. So in this way they catalyze oxidation and reduction reactions. Examples of oxidoreductases: dehydrogenase, reductase, oxidase, and peroxidase. Okay, now let's see transferases. The name itself tells that they are involved in transfer of something, right? They catalyze the transfer of groups from one substrate to another substrate. Let's see the mechanism of reaction. This is a substrate S attached to a group. S dash is another substrate. The transfer is simply what they do. They transfer the group from one substrate to another group, because of which you can see here S is left without any group and S dash is attached with a group. Understood? Transferases examples are transaminase, transketolase and kinase. Okay, next hydrolases. These are one of the most important enzymes and everyone knows them. These enzymes we find in our digestive system. They help in digestion of our food material by a mechanism called hydrolysis. Okay, the enzymes which break down carbohydrates, that is carbohydrases, the enzymes which break down proteins in the digestive system, that is proteases, and the enzymes which break down lipids, lipases, and the enzymes which break down nucleic acids, nucleases. All these belong to this category called as hydrolases. Okay, let's see. They catalyze hydrolysis of ester, ether, peptide, glycosidic bonds and carbon-carbon bonds, carbon halide bond, PN bonds by adding a water molecule. So what is common in all these hydrolysis reactions? Water molecule is added, then the bond is broken. Okay, that's why it is called, these enzymes are called hydrolases. If you see the mechanism of reaction, let's see A and B are attached by a bond. Now by addition of a water molecule here, the enzymes will break the bond. So A gets separated from B. Now A is attached with a proton, B is attached with a OH group. So this is the mechanism of reaction catalyzed by hydrolases. Examples, your digestive enzyme amylase, pepsin, another digestive enzyme, and nuclease. Okay, now let's see lyases. Lyases, they remove the groups from substrate by mechanism other than hydrolysis, by leaving double bond. So one of the important character is they leave double bonds after removing the groups. Let's study the mechanism of the reaction catalyzed by lyases. Let's see here, two carbons are attached by bond, carbon-carbon bond. One carbon is attached to X, another carbon is attached to Y. Now, these two groups should be removed. So what they do is, they simply remove the groups from carbons, both the carbons, by breaking these two bonds and attach the two groups. When two groups are attached, then the two carbons are left with double bonds. So this is the mechanism of the reaction conducted by lyases. Examples of lyases: aldolase, decarboxylase, enolase and hydrases. Okay. Next, isomerases, the name itself tells that they are involved in isomerization reaction. Okay, they catalyze interconversion of isomers such as optical isomer, geometric isomer and positional isomers. Let's see this molecule S dash. One side there is X and another side there is Y. This isomerizes, simply they will change the position of the groups. You can see X has been transferred its position from upside to downside. So this type of reactions are catalyzed by isomerases. Example of isomerases are hexose isomerase, triose phosphate isomerase. Okay, can you tell in which process this hexose isomerase is involved? Glycolysis. Triose phosphate isomerase, it is also involved in glycolysis. So I have given the examples, both the examples from glycolysis. Okay. Now let's see ligases. Ligases, they link together two compounds. They link together two compounds by formation of bonds. They can link carbon oxygen, carbon sulfur, carbon nitrogen, phosphorus oxygen by creating bonds. If you see the mechanism of reaction, A and B are two substrate molecules. Now with the help of ATP energy, bond will be created between A and B. You can see here A and B are linked together. During this bond formation, ADP and inorganic phosphate PI will be released. Example of ligases are DNA ligases. These enzymes are playing a very important role in DNA replication process where these enzymes will join the DNA fragments together by creating ester bonds. Okay, so this is about the classification of enzymes. Copy down, students. Now we are going to discuss the added syllabus by NTA. NTA has added this particular topic called as properties of enzymes. So I am going to discuss with you 8 properties of enzymes. First property, catalytic property. You know that the enzymes catalyze the chemical reaction, they increase the rate of reaction. The enzymes can speed up the rate of reaction without changing their own original structure. Okay. Next, specificity. Every enzyme is specific to a particular chemical reaction. Okay. Enzymes catalyze only specific reactions. So this is called as enzyme specificity. Next is reversibility. You know that most of the enzymes can catalyze the reaction in both the directions, forward direction and backward direction. This is called reversibility. Most of the enzymes can catalyze a reaction in either directions. Okay. Next is sensitive to temperature. Sensitivity to temperature. Enzymes, of course, they are protein molecules which are sensitive to temperature. They get easily affected by change in temperature because change in temperature will affect the tertiary structure of protein. Okay, it will affect the bonds, hydrogen bonds and sulfur disulfide bonds. Hence, there is sensitivity to temperature. So most of the enzymes undergo denaturation above 40 degrees centigrade and they get inactivated at freezing point, that is 0 degree centigrade. So look here, the enzymes, most of the enzymes undergo denaturation, means they lose their tertiary structure above 40 degrees centigrade irreversibly, and the enzymes get inactivated, just inactivated at freezing point of 0 degree centigrade. Okay. So there is a difference between denaturation and inactivation. Denaturation means permanent loss of catalytic activity, whereas inactivation means temporary loss of catalytic activity. Got the point? Now sensitivity to pH. Enzymes are also sensitive to pH. Okay. Enzymes, every enzyme has its own optimum pH. That already we have discussed in factors affecting enzyme activity. Enzymes denature at very low pH and also at very high pH. Next, protein nature. Enzymes, of course, they have protein nature. They are globular proteins with high molecular weight. Okay, and that is the reason that enzymes will not be transported from one cell to another cell. The main reason is that they have high molecular weight. So every cell will synthesize its own enzymes instead of depending upon some other cells. Got the point? Now, chemical reaction. They do not start the chemical reaction. Enzymes do not start the chemical reaction. They will just act as a catalyst. Got the point? And they do not change the equilibrium of reaction. Next, turnover number. Enzymes have turnover number. What is turnover number? The enzymes can convert a specific number of substrate molecules. They can convert specific number of substrate molecules in unit time. We can also call it as efficiency of enzyme. Okay. For example, carbonic anhydrase. This enzyme is the fast enzyme. It can catalyze many reactions. It can convert many substrate molecules into products in a unit time, whereas lysozyme is slow in catalyzing the reaction and it has low turnover number. Already in the beginning of the class we have discussed about carbonic anhydrase. Do you remember? 6 lakhs molecules of carbonic acid are produced per every second. So that is how fast is carbonic anhydrase. So with this, successfully we have completed the chapter Biomolecules. Let us celebrate. So here, I am not discussing the deleted topics for NEET examination. You can see here in page number 152 from Biomolecules, dynamic state of body constituents, concept of metabolism is deleted. So we are not discussing this. Metabolic basis for living, this topic also is deleted for the NEET examination. The living state, this topic is also deleted. Okay. So Students, all the best. Prepare well for your NEET examination and always dream of achieving your goal, that is getting a seat in topmost medical college. All the best. We'll meet in the next video. Subscribe to this video and also give a like and also do comment. Thank you.